myself Nikita, and I'm from University of Delhi. So today I'm going to present on the role of pro-sequences in protein folding and functions and structural insight. So what actually are pro-sequences? Pro-sequences are stretches of amino acids which are translated protein, and they are later filled upon their functions. So the first one to be reported was success from the Bacillus subtilis, and later on there are various proteins were reported to have pro-sequences. So what pro-sequences usually used to do? Pro-sequences usually was first reported to help in protein folding and then cleaved off. So they were termed as intramolecular chaperone also. So we have a database called ProSeq APBJC, which is made in our lab only by me and my guide. So it has been reported to have 2,140 pre-protein containing assisting proteins. These proteins are called as pre-pro proteins. So these pro-sequences generally affects the folding of the cognate protein, but later on it was thought to perform other functions also, like they help in signal transactions, enzyme targeting, as well as preventing the protein from the other proteases in the cell. So what our aim of the study? We wanted to see like how pro-sequence modulate the structure of the protein, and further how ligand binding sites are modulated, and what residues are basically involved in the pro-sequence functions. So what we did here, we took the pro-sequences assisted proteins from the database containing N and C terminal pro-sequences, because they are mostly found at N terminal, and some of them are found at C terminal as well. Some are reported to be, present in between also, but here in the database we have taken the terminal pro-sequences only. So N terminal and the C terminus pro-sequence containing proteins were taken. From there, we did a blast search to see blast search against the PDB structure database, so to see how many PDB structures are available against the database. So upon that, we took the 100% coverage sequences, because we wanted to see what is the role of pro-sequence. So we took 100% coverage sequences along with 30% sequence similarity, minimum of 30% sequence similarity, and then we did a residue interaction network analysis to see there is a residue interaction between the residues of pro-sequences as well as protein. Further, we did a pairwise alignment to check the alignment of PDB structures as well as the pre-pro proteins that we have taken previously. Then we did a like ligand binding pockets analysis to see what are the ligand binding pockets which are available there, and then we did PDBSim for ligand binding residue analysis. So upon the study, we got to know that there are 127 PDB structures for N terminus and 14 structures for C terminus available from PDB. And as we can see here, like only for N terminus, P proteins and PDB pro-sequences protein interaction was seen. So we did not take the C terminal data further, and later we found that 14 Cas-P like binding pocket information was available, and for PDB some only 12 have ligand information available. From there we can see that there are interactions involved. So if you can see there what type of interactions we studied. For the one residue is the amino acid of the pro-sequence involved in the binding pocket, and the second one is the protein, as well as protein is directly in and it is helping in binding the pocket. And the third is the binding pocket residue is in contact with the protein residue, and which is in direct contact with the pro-sequence. So this way we got into study. So here you can see there are 12. There are in total 14 structures like that from the Cas-P, and from PDB-SUM there are only 12 P proteins to have this interaction. So here you can see there are different number of PDB structures with the residue information. For more details, you can come up to the poster as well. So it was seen that not all pro-sequences are in direct association, because there were 735 sequences we have for N terminus, but only 127 were reported to have PDB structures. And then there is interaction of pro-sequence in forming the binding pockets, as well as there are some pro-sequences residue to be involved in ligand interaction directly. So in future we'll try to see the exact conservation of the residues among other binding pockets, and we'll try to do it with the help of simulation to how mutation can really affect the binding pocket interactions. So yeah, Thank you. I would like to thank my entire lab mates, as well as the department, as well as the funding body UGC, CSIR, ICMR, and Delhi University. Thank you. For any questions.